Hi. In this short video tutorial, I'm going to show you how easy it is to analyze differentially expressed genes in your bulk RNA-seq data. Here, I have some paired FASTQ files already added to the project. This is bulk RNA-seq data, and I have some metadata attached to those samples, and you can see here we have column which is called treatment, and we have three groups of samples: control, geo, and GOC. Now what I want to do, I want to find out what genes are differentially expressed between geo and control and between GOC and control. Let's do that. So the first thing we're going to do, as we usually do with any kind of RNA-seq analysis, we need to align our transcriptome data to the reference. And for that, I'm going to use the block which is called star. And this block uses the software of the same name, star, which is one of the most commonly used tools for RNA-seq alignment. So here in the settings, I'm just going to select my data set. I select the species, in this case is this one, and select the strandness of the data. In this case, it's unstranded data. And I'm just going to click run. That's all you need to do. And now the reads for the RNA-seq data are being aligned to the reference. And you can see the progress here, just as with any other blog that runs the software under the hood. You can see the logs for the software running. And it will just take a few seconds to finish, and we will have the results. So now right away you see 3 samples already done, 4, and that's it. So now we have all of the samples processed. We see a high read alignment rate, which is very good. And right away, without even deep diving into anything else, what we can do here, we can look at the principal component analysis, the PCA. And here you see that we already kind of see these samples. We have 6 samples in groups of 2. They already kind of cluster together. Let's go and check if they do cluster based on the treatment conditions. So I'm going to drag this treatment to the grouping color. And yes, they do. So we already see that we have a group of samples here, here, and here. So these 3 groups clearly show different pattern of gene expression. Now, the next thing that we want to do is to actually evaluate which genes are differentially expressed. And for that, I'm going to go and add another block, which is called differential expression. And under the hood, this block uses one of the most commonly used softwares, which is called DSeq2, to analyze differential expression. In the settings, I have to select the dataset, which is RNA-seq. Now I have to select the design, and design is a factor which you assume has an effect on the gene expression. In this case, I'm only going to select treatment because that is what I think. But sometimes you have a more complex metadata and you think that many factors affect the expression of your genes. So you might select several columns here. So design is what affects the expression. Then contrast factor is a name of the column over which you want to compare the differential expression. So in my case, it's just treatment. And what I want to do, I want to compare the differential expression of the genes between GOC and GO over the control group. So what it means, if I want to separately know how genes are differentially expressed between GOC and control, GO and control. That's all that I need to specify. And I'm going to click run right away. And DSeq under the hood will generate the table with log2 fold changes for all of the genes. So here I see the list of the genes and the values, the log two-fold change values for the expression between GO and control, or I can change and look at the other group, comparison GOC versus control. And one of the most commonly used ways to look at the differential expression genes data is a volcano plot. And we have it right away here. So I can click on it, and you see we have the volcano plot that shows us that between those two groups, GO and control, these are the genes that are upregulated, and these are the genes that are downregulated between those two groups. Now, what if we want to look at a certain gene, like at a certain specific gene that like maybe you don't see at the volcano plot? You can go at this single gene level if we add gene browser. So I'm going to add gene browser to my project and select normalized gene expression, because when we want to compare the expression of genes between groups, we need to normalize data. So here I have samples and genes, and I can go here. And for example, let's say I want to look at this gene. So this is the expression of this gene, and now I also want to compare the groups in my treatment column. So I'm going to go and just move treatment to primary grouping. And right away, now you see the expression, the level of expression of this specific gene over 3 groups of samples that I have. And you can see how it changes. Now you can change the order of this group. There's many ways how you can change the visualization of this plot. Like for example, like maybe I want the order to be different like that. Everything can be done, or I can add some statistics if I want to compare these values of expression, anything. Now, finally, what I also want to do, not only I want to know the expression level of single genes, but I also want to know the functional analysis. I want to know what pathways are enriched or downregulated between my groups. And for that, I have a block which is called functional analysis. And let me add this to the project. And in the functional analysis, again, I'm going to just select my data set. I can select the collection of the pathways that I'm going to use. For example, gene ontology, again, one of the most common ones. This I can leave as is, upregulated and downregulated. And I'm just going to click run and see what it shows me. So now this is, again, another way of looking at the genes. So we looked first at all of the genes on the volcano plot. Then we, using gene browser, we looked at the expression of a single gene. And now we want to group those genes into pathways and see what is changing in terms of pathways between my groups of samples. So let's wait for the analysis to complete. And here we see the pathway ID and the adjusted p-value of the change. And here, if I click on the bar plot, now I see the downregulated pathways between GOC and control. I can do the same for upregulated. And now you see that for upregulated, I have actually upregulated genes in GO versus control and GOC versus control. So this is an easy way how you can analyze the changes in the pathway expressions between the groups of your samples. That is all that I wanted to show you today. Thank you for watching.